Okay, thank you so much for the very nice introduction. I'm totally thrilled to be here. And today I'm going to talk about function annotations and their application. We've learned a lot about GWAS in this week, but this talk starts with GWAS limitations. So first of all, GWAS is a massive multiple testing problem because in your tutorial, you've already seen that we study millions of markers simultaneously, and you need to correct for p-value. And we already know that for most human complex traits, the effect size is actually pretty weak. So you have weak effect size, and you have a massive multiple testing problem. That's why you need extremely large sample size to identify all those signals. And Johnson Pritchard and colleagues at Stanford has a very nice perspective article in Cell that came out a few months ago talking about this. So basically, we're facing not just polygenic architecture, but sometimes even they call it omnigenic. So it's a big computational and statistical burden in GWAS analysis. So before that, I'm just going to talk a little bit about the outline. This is the outline I'm going to follow. I guess I'll just quickly march through a little bit of background of GWAS and GWAS limitations, and also the function annotations in protein coding genes. But then I'll focus more on function annotation in non-coding regions and their application in GWAS analysis because it's more closely related to our topic here. Okay, let's go back to the background. So challenges. So due to polygenicity and low effect size, GWAS has been controversial. And even if you are able to identify GWAS association, it remains very challenging to interpret these findings. And one of the reasons is that nearly 90% of significant associations are actually located in non-coding regions, the part of the genome that we don't fully understand yet. And another reason why this is challenging is because we have this phenomenon called LD. Right? LD is really the basis of GWAS, but at the same time, it harms the interpretation. Because basically, by doing GWAS, you get some rough idea about where the signal is, but fine mapping of truly functional variant is extremely difficult. So here are two examples to illustrate the idea. They are from a COPD GWAS. You can see this is a big LD block. It's genome-wise significant, but it's in the middle of nowhere. There's no gene down there. So how do we interpret this? What exactly is driving this association? It's difficult to say. Here is another LD block. This time it's the gene-dense region. So although we have some gene candidate, but it's not trivial to figure out which one of these, or maybe multiple of these genes, are truly driving the association. So we have problems. If we can understand better what this region does, that might be a starting point to interpret what GWAS tells us about the complex trait we study. Okay? And to solve all these problems, we need better function annotations, especially in the non-coding region. And just to give you some sense about human genome, someone found a printed copy of human genome. And this is how many books you need. And within each page, it basically looks like this. That's how massive the data is. What we are trying to do is to circle out certain elements to say maybe this is a protein coding gene. This is relatively easy. Maybe this is an enhancer to a nearby gene, or this is something we don't fully understand yet, but we have empirical evidence suggesting that this may be related to neurological function. So piece by piece, we will try to annotate the human genome and use that information to help us understand what GWAS is telling us. Yep.
Can you just define functional annotation?
It's a very broad concept, and I'll actually come back to this point later. Different people will have different image in your head when we talk about function annotation. So I'll start from very narrow sense function annotation prediction tools, basically given a SNP, how likely it is going to have a biological impact. We'll start from those tools, but then I'll try to broaden that concept into something else. Basically, to me, function annotation equals to external information. And I'll try to address that later. Okay? And we know that 98% of the genome is non-coding. But overwhelming evidence suggests that they may be critically involved in all sorts of complex trait mechanism etiology. Okay? All right, so let's take a look at the narrow sense function annotation prediction tools. I'll start from protein coding genes, which is relatively easy, because we have better understanding of the protein coding genes. So basically, you have, in protein coding gene, you have this concept of codons, right? It's a 3 nucleotide corresponds to 1 amino acid. And sometimes, it's not one-to-one correspondence. Different codons correspond to different, amino acid, but it's not one-to-one. So you may have a variant in a codon, but sometimes it actually does not change the amino acid sequence, and that's called synonymous variant. So those variants are unlikely to be very harmful, but sometimes you do have changes. For example, if you mutate A into C, then actually changes the underlying amino acid sequence. So this type of variant is more likely to be functional in some way compared to synonymous variant. And there are more damaging variants compared to those types. So there is this thing called stop codon. Basically, whenever you see a stop codon, translation stops. Okay? So if your variant makes amino acid that's not supposed to be a stop codon, but turns it into a stop codon, then you're going to have a problem because the protein product is going to be shorter than the normal form. So it's more likely to be damaging. And sometimes if you have an insertion or deletion, it just frame shifts the whole thing, and the amino acid becomes random. And those are also more likely to be damaging. And some of these are summarized as loss of function variant. So these are more likely to be damaging than just missense variant. But still, it's a non-trivial problem to tell how likely a missense variant is going to have impact on biological function. So computational tools have been developed. Usually, they are based on supervised learning methods. Popular tools include SIFT, Polyfan, and MetaSVM. Just to try to use this to illustrate what's the idea of supervised learning. Basically, you need to use examples to teach your algorithm. You need to give a lot of examples for Russian blue and a lot of examples for Maine Coon. And you need to throw in a bunch of predictive features, for example, the length of hair, the color of eye, and something else. And the algorithm need to figure out how to distinguish these two categories. And then when given a new figure, the algorithm will tell you how likely this is a Maine Coon, how likely this is a Russian blue. It's exactly the idea what people used in supervised learning methods for figuring out whether or not a variant is functional, right? So because in its coding region, we have databases that tells you that gives you a list of disease-causing variants, and you also have a database for relatively neutral variants. You compare those two categories just like you compare Maine Coon and Russian blue. And we also have predictive features. You know how likely they are going to change the protein sequence. And you also know sometimes if you change the amino acid, it also changes the protein structure. Those are the predictive features, and there are well-established algorithms that helps you distinguish how likely this variant is going to have an impact. So this is a rough idea. And just one quick application. Why is this helpful? So this is slightly different. So people have been doing this analysis for de novo mutation. It's just very quickly, it's very intuitive. So parents are actually non-carriers for the mutation, but sometimes in baby, you see mutation. On average, everyone in this room will carry 60 de novo mutation in the genome, and roughly 1 will hit protein coding regions. And if this mutation hit a critical gene, then you are likely to develop some kind of disease. So then you can kind of, we have a very good idea about how big the mutation rate is. So we know how many mutations we expect to see in, for example, 3,000 family trees. And then we just compare the expected number of de novo mutation versus the observed number of de novo mutations. There's no enrichment. But if you focus on damaging mutations, there's a massive enrichment. So without annotations, this kind of signal is not seen. Basically, if you pull together the damaging mutation and non-damaging mutation, pretty much dilutes the signal. But if you just focus on functional variants, the enrichment is actually very significant. And for protein coding regions, transcription is another type of function annotation. So if you just this is for congenital heart disease. If you just focus on genes that's specifically expressed in a developmental stage for heart development, then the enrichment becomes even more massive. If you do exactly the same thing in control trials, you don't see any enrichment. So this is just an example trying to convince you that function annotation is powerful if you use it properly. Okay? But more importantly, we want to study the non-coding region. So what about non-coding region? What kind of data can we use to help us figure out which region or which variants are more likely to be functional? So one type of data is DNA conservation. It's based on multi-species alignment. You compare the DNA sequence across different species, and if the DNA element is very conserved across different species, it actually hints at functionality. Because basically, it tells you that natural selection has wiped out all the mutated versions. It means this region must be important, right? So people have done computational methods to quantify that. So that's one type of information you can use. Another type of information is transcriptomic information. So as long as this region is transcribed, it already gives you some information, right? So transcribed region is more likely to be functional than things that are not transcribed. But if you combine this transcription information with genotype information, you can also do EQTL analysis. So even if this specific region is not transcribed, but the SNPs at this locus is associated with differential expression for a gene, then it's a type of function annotation for the SNP you are studying, right? So a non-coding RNA and EQTL are two types of things you can derive from transcriptome information. And there is also epigenetic information. So the DNA sequence, if we ignore somatic mutation or mosaics, then the DNA sequence is actually the same across all cell types in your body, right? But different cell types are doing very different things. And what's the reason? So part of it is due to epigenetic regulation. So basically, your body has this very complex regulatory machinery that controls this cell type. Different part of the DNA sequence is turned on or off in different contexts. And that's called epigenetics. So we now have high throughput technology to measure whether or not this specific element is open, for example, in microglia. And maybe it may not be open in neuron cells. So that type of annotation or data can certainly help you figure out. It's certainly a useful feature to help you predict whether or not specific variant is functional. Okay, so these are the type of data you can use. And then people have developed functional methods. Once again, we started from supervised learning. You feed in examples and try to figure out what's going on for the new unknown variant. But the problem is that in non-coding region, we don't have a very good understanding. We don't have a very good database for functional or non-functional variants, right? If you mix the dog's picture in this algorithm, it just confuses the algorithm. And if you have a Maine Coon in the cluster for Russian blue, once again, confuse the algorithm. So supervised learning is not immune to noise in training data. If you don't have good understanding for regulatory variants in the human genome, you can't train the algorithm very well. But there are methods. One is called CAT, developed by Jay Shanduri at just UW, and GWAS is by Paul Flichak from the UK. So, but there is another type of function annotation tools. It's called unsupervised learning. The idea is different. Basically, you're throwing all data you have, and you try to let the algorithm learn certain features from the data and cluster them into different categories. It's kind of like the PCA analysis we did yesterday, right? So the problem of this type of approach is that there's no guarantee what you're going to get in the end. You have to interpret that, and it's a non-trivial problem. If you only have two clusters, you may just assign one as functional, another is non-functional. But when you have 5, what's going on there, right? So it's a non-trivial problem. Yes?
Is this also the algorithm is a black box, so you don't really know what exactly it is?
Depends. You can use model-based approach. You can use parameters to make sense out of it. But in the machine learning world, people believe that black box algorithms actually performs better. But there's a trade-off. You lose interpretability. Okay? So, and there are methods. We actually developed some of the methods in this category. We have GenoCanyon, GenoSkyline, GenoSkyline Plus. The difference is that GenoCanyon is non-tissue specific. If you use things like transcriptome or epigenetic information, the raw data is tissue specific. If you only use, for example, microglia data to train the algorithm, the outcome is going to be microglia specific, right? So we started from non-specific to more and more specific. And there are other algorithms too. So Eigen is from Columbia, and Chrome-HFM is from Broad Institute. Okay, so let me know if anything is unclear. Okay, and just a few other useful tools. UCSC Genome Browser. I see Paul is wearing a UCSC shirt today. It's the most useful tool to visualize all these annotations. To basically input a genomic region, and you can manually select which annotation you want to see. It's not very quantitative, but it's a useful tool. And there's another one called ANOVA. Actually, it has incorporated many of the computational tools in its pipeline. You can just install it and input your VCF file. It automatically helps you extract the annotation. It looks more boring. Actually, it's more useful in your data analysis. Okay? So the more exciting part of this is, so we've already had a long list of annotations. How exactly can we use this information to help us understand complex trait? Specifically, how do we integrate this information with GWAS or just GWAS summary statistics to make sense out of a result? So I'm going to give you several different applications. The first one is just functional SNP fine mapping. So once again, just the example, right? a large LD block in the middle of nowhere. What's going on? Which SNP is more likely to be functional, and then how do we study its function? And there are several different methods that can do this. These are called fine mapping tools. One is Painter from UCLA, GWAS is from Joe Pigura from New York Genome Center, and we actually have another algorithm called GWAS, Genome-Wide Association Prioritizer. The intuition of all these tools are very similar. We're not going to get into the mathematics, but the intuition is that in the end, you will get some kind of probability that tells you how likely each variant is causal, and the input information is p-value or summary statistics from GWAS and the underlying annotation. Right? So if you throw in a bunch of annotations, these p-values are all very similar because of LD, but if one SNP is located in an extremely functional region, of course you want to push the signal up. If the others are located in non-coding region, non-functional region, you want to drag down those signals a little bit, right? The problem is just quantitatively, how do you do that? That's why people have proposed different models. But the intuition is actually pretty clear. So specifically, how we did that is that if you have a very rough functional annotation for the genome, and you compare the p-value distribution between SNPs in functional region versus SNPs in non-functional region, you see a difference, right? P-value is slightly higher, the peak is slightly higher in functional region. But importantly, you also see a peak in non-functional region. That's exactly what LD is about, okay? But the difference actually tells you about the behavior of truly disease-causing variants, okay? So basically, it's based on mixture model idea. You want to remove this part, because this is just telling you how much impact LD has. You want to remove this part from this distribution, and the remaining part is the behavior of disease specific functional variants. And of course, using some mathematics, you will be able to get a posterior probability of being causal given annotation and p-value. And that helps you prioritize GWAS associations. So to give you a better illustration or visual impression, basically we start from summary stats, integrate with annotations, and reprioritize everything. You can see the peak actually changed. And turns out we compared the prioritized results with this is for COPD, so we compared it with the 3C data set in a specific lung cell type. Basically, this type of data tells you the physical interaction between different loci on the genome. So it turns out this is the only probe that has a strong interaction with the promoter of HHIP, the only nearby gene. So this is indeed an enhancer candidate. And people have done follow-up. This has been validated. So this is just a toy example to show you how this type of tools may help you. But if you go to this specific SNP, it's not an enhancer candidate, and just maybe it's not that easy to see. So this specific SNP on this plot is right here. You can see even the p-value is not that impressive. Okay? So one example. So the next example is about partition heritability and genetic covariance. I think in the polygenic risk score talk yesterday, Emma talked about LD score regression. So that's exactly what I'm going to talk about to give you more details right now. So the idea is that after you do GWAS, you get a p-value or summary stats for every SNP in the data set, right? The thing is, before doing that, you know where the SNPs are. And you know some region has higher LD, some region has lower LD, right? The whole point of LD score regression is that you actually expect to see stronger association in a region with high LD. The intuition for that is that if you just randomly throw 100 causal SNPs in the genome, a region with high LD is more likely to just tag that one of the causal variant, right? So let me know if this is unclear, but I think it's an intuitive idea. But what the very nice thing about LD score regression is that under reasonable assumptions, the proportional relationship between summary statistics and LD is actually linear. So you can simply run linear regression and estimate the parameters. And even cooler, the slope is heritability. Okay? So just with high LD, you expect to see stronger signal, and you just regress those two variables. The slope can be interpreted as heritability, which David Fardo talked about the other day. That's the coolest feature of LD score regression, but why is this useful? I'm not a big fan of just purely heritability estimation. I don't really think it makes a lot of difference whether this is 40% or 55%. I don't think that difference is that important. But relative difference is important because you can essentially extend this to incorporate functional annotation in it. Okay? Now you have two parts of the genome. One part is brain-specific functional region. Another is regions that are not functional in brain. Then you can estimate what percentage of heritability is located in this part and what's the percentage in the remaining part. Right? Suppose your brain-specific functional region covers 10% of the genome. Then if there's no enrichment, you expect to see 10% of heritability to be located in brain functional region. Right? But if you observe 30%, then there's a threefold enrichment. You quantify that into statistics and you get a p-value, how strong the signal is. That's essentially what this is. So enrichment is the ratio between percentage of heritability explained by SNP in that annotation divided by the percentage of genome covered by that annotation. Okay? And if we do that, basically we can analyze GWAS traits. Right? Once again, the intuition is you identified a lot of SNPs for Alzheimer's, schizophrenia. You can map them down to the genome to see which annotations they are located in and use this enrichment to calculate how strong, to quantify this kind of enrichment. And interestingly, for schizophrenia, we are able to see that it's massively brain enrichment. But for Alzheimer's, it's not brain at all, but you see a massive enrichment bar for blood tissue, which is largely representing immune cells. Okay? But we have a roadmap project did a good job by providing high-quality data for more than 100 different tissue and cell types, and we did exactly that. So basically, we can do much better than the previous 7 tissues if you refine this into, in this specific example, we use the 66 different annotations. This specifically highlights monocytes for both AD and Parkinson's disease. And just as a comparison, if you look at the enrichment for ADHD, which is a psychiatric disease, you can see the enrichment actually in brain region, specifically anterior caudate, but not in other tissues. So you can potentially do similar things using transcriptomic data, but my experience is that it's much more robust. If your goal is to infer disease-related tissues, it's much more robust to use epigenetic information. And I'll get back to this point just a little bit later when I talk about things like predicts. Okay? And for AD, by the way, there's another signal in liver, which may be representing lipid metabolism. Okay? And
if you remove ApoE, the ApoE
liver is still significant. We did that. And Alison has
So our data is the same as yours, right?
Okay. Yeah, we saw that paper.
And liver is the top 2.
Yes. And I think the slight difference is that we specifically highlight monocytes, and the trick is that because there is similarity between different annotations, so it's kind of like LD across different tissues. If you do it one by one, like in GWAS, you analyze SNP one at a time, you see a lot more enrichment. But if you actually do a multivariate analysis by throwing in a large number of tissues, the only one that remains is monocyte. Okay, so yeah.
Sorry, can you just recapitulate? So what does it mean that monocytes has more enrichment when it comes to ADs? That means
so I'm just making up specific numbers. But for example, for AD, IGAP SNPs can explain, let's say, 10% of heritability. That's 10% of phenotypic variability, right? That 10% is called heritability. Now, within this 10%, a large proportion actually comes from monocyte functional region. If you divide that proportion by the actual physical proportion that this annotation region covers, it's a huge enrichment. So that's what I'm talking about. So in fact, if you add up monocyte functional region and liver functional region, virtually explains all the heritability for Alzheimer's. Once again, it's not saying that they completely explain phenotypic variability for Alzheimer's. It's given the heritability, everything comes from these two categories. Okay? All right. Yeah, and we also see that in Parkinson's disease. Okay, so that's consistent with many of the recent reports about the importance of myeloid cells. But one critical limitation is that we don't have data for microglia. So it's totally possible that this is just epigenomic similarity between monocytes and microglia. But I think people haven't really ruled out the possibility that monocytes can be directly recruited through the blood-brain barrier to participate. So it's a non-trivial problem, and it's an open question. Okay, the next topic I'm going to talk about is genetic covariance. So before, it's one trait analysis, right? We want to figure out where the variability comes from along the genome. Turns out we can also study that for a pair of diseases. We know that, for example, schizophrenia and bipolar disorder are significantly correlated, but is that correlation coming from brain functional regions? That's a type of question we can answer. So once again, LD score regression. One of the LD score regression papers did this genetic correlation method. The idea, basically, it's a metric that quantifies shared genetics among complex traits. I'm not going into the mathematical details, but the idea is that SNPs have genetic effects on the first trait, and they also have genetic effect on the second trait. Those two random variables, there is a covariance parameter. We want to do inference to see that covariance is 0 or non-zero. Okay? That's what this is all about. Okay.
Hold on. That's very interesting because in Alzheimer's there could be some pathways that have shared variants with the, say, amyloid biomarker and, say, a cerebrovascular disease variable. So is that the kind of thing that you're talking about here, that you could, that these two complex traits, you could map the shared genetic?
I would say this is a starting point. There are some technical issues because for biomarker GWAS, typically the sample size is smaller. This actually requires a sample size to be over 5,000. Another technical difficulty is that for that specific example, you're probably more interested in causal relationship. You want to see which one is affecting the other, right? Just correlation is not satisfying enough. But this doesn't give you that. Okay, but there are people who develop a new method based on this similar idea and try to answer some of that. But so far, I mean, it's still largely open question. Yes. So does that answer your question?
Yeah.
Okay. Yeah.
Just trying to understand what it is that you're saying.
Yep. Uh-huh. And the interpretation is very tricky. For example, we are able to see a significant negative correlation between being a morning person with education attainment. So I think it's a funny example. But the interpretation is tricky because I think Amo also mentioned some of that the other day. So it could be because there is a directly causal relationship between being a morning person and education attainment. I mean, being a night person means you maybe works a little bit harder. I mean, it's controversial, right? But if that's a thing that leads to lower education attainment. But it could be the other way around. If you don't get enough education, maybe it forces you to get certain jobs that makes you a morning person, right? So it's a mess when you interpret those things. Yes.
Or that you've got some sort of personality.
Yes. It could be all sorts of confounders. Yeah. Exactly. But in disease genetics, it's relatively cleaner. Okay. All right. So the point is, we have a new statistical estimator that's better than LD score regression. I just marched through this. Basically, both our method NOVA or LD score regression are unbiased, but our variance is smaller, and that translates into higher statistical power. Because whenever you see a non-zero value, the variance is smaller, so you have more statistical evidence saying that this is actually indeed non-zero. Okay? So ours is more powerful, and we can do annotation stratified analysis. LD score regression cannot. So basically, we studied late-onset Alzheimer's disease versus amyotrophic lateral sclerosis. We tried to dissect the genetic architecture, the shared genetic architecture between these two diseases. We can see that, first of all, LD score regression gives you a p-value of 0.07, which is not impressive at all, but our p-value is 2 times 10 to the negative 4. And more importantly, almost all the covariance come from predicted functional region. So if you just focus on predicted functional region based on our annotation, the p-value is even more significant. And you can also partition the data set by minor allele frequency. You can show that the covariance is mostly explained by common SNPs. That's interesting because, rare SNPs are enriched for heritability for both diseases, but it turns out the shared genetics is from common SNP. So those are the type of questions you can answer. And you can also partition the genome by chromosome. I mean, why is that interesting? The reason why is if the shared genetics between ALS and Alzheimer's is polygenic, basically if the shared genetic components are randomly distributed alongside the genome, then you expect to see a linear relationship when you plot chromosome size versus the value of covariance on that chromosome, right? But interestingly, we don't see that at all. But remember, in the previous slide, I showed you that all the covariance come from predicted functional region. So if you just remove the non-functional region and plot the size of functional region on each chromosome versus the covariance explained by SNPs in functional region, now you see a linear trend. So it's actually polygenic. The shared genetics between these two traits are polygenic. But if you don't reduce noise, if you don't remove non-functional regions in your genome, it introduces a lot of noise and basically hides the signal. Okay, that's the point.
Okay. So based on the GWAS data that we have right now, I don't think there are any genes in common that were genome-wide significant, right? So that's suggesting that it's all in the noise right now where we have the overlap?
You mean for ALS?
ALS and the
Oh, they have a recent study. I actually didn't notice if they have a genome-wide significant locus, but it's not that surprising because the idea of this is that it's polygenically correlated. So we're talking about very weak effect size, but they tend to the direction tend to be consistent. So yes. So I would say first of all, it tells you about the genetic architecture. It doesn't have a low-hanging fruit, and in the currently non-significant loci, in the non-significant genome, there are definitely signals. When you further increase sample size, you will expect to see significant hits. Because biologically, it makes sense.
So if you use Genova with the ALS data,
yes.
epigenetics, what comes up in your traits?
Good question. We did that. So immune, 0.01. But it's non-significant after multiple testing correction. P-value is 0.01. The sample size is actually not too bad. So it's quite impressive. To my knowledge, it's the most recent study for ALS. Yeah. Yes?
I think this approach is really fascinating to get at this question of like disease mechanism or mechanisms that are not disease specific but are important for neurodegeneration and aging. And it seems like this should exist to some level, and you're really able to kind of accept that with this approach.
Mm-hmm.
So I guess one question is, can you do this with multiple diseases? Like, could you take various summary stats and across the different neurodegenerative diseases and understand
So we haven't implemented that yet, but technically you can. Basically, we're talking about the correlation among genetic effects, right? So if you throw in more diseases, it's going to be a network. Yeah. So theoretically you can do that, but there are all sorts of technical difficulties because even by analyzing a pair of disease, you have to remove the contribution due to shared sample size. Shared samples, that's a non-trivial problem. If you have 3 or 4 diseases, it makes the problem even more challenging. So I'm not sure if there's a good way to solve that particular technical problem. But if everything is clean, data sets are completely independent, I wouldn't expect this problem to be too difficult.
And then I guess another technical question is the controls are different, right? In each summary stat file that's going into it.
Actually, they're always overlapping. So the control part is the overlapping part because everyone uses WTCC controls as part of the control set.
Well, not everybody, but I mean, there's actually in the neurodegeneration.
Oh, because they have to test that. I see.
But there is a problem. If you look at like the PSP GWAS and the MTE GWAS, they all use the same controls. So
yeah, but mathematically we can demonstrate that if your design is different cases but completely shared controls, this is still valid. It's part of our paper. But you can have the situation where if this individual is selected to be cases for first study, then actually it's more likely to be selected as a control for the second study. You can't have this reverse relationship. That will ruin your analysis. But you can share controls. That's not a problem. Yes?
I think this might be a partial answer, at least to the best question. Broad released MTAG, some multi-trait analysis.
Oh, yeah. Yes.
I don't know the details of it well enough.
Yes, I think their point is to estimate effect size. It's not about testing genetic correlation. Basically, if you have 5 GWAS for all sorts of psychiatric traits, can you better estimate effect size for all of them? And I'll actually talk about that a little bit later on. Not exactly that method, but similar ideas. Yeah.
Was it this paper where they had psychiatric phenotypes as well as some of the neurodegeneration ones? And in psychiatric disease, you see a lot more genetic covariance
than neurodegeneration. Yes, AD is on this figure somewhere. Oh, yes, this one. So nothing was significant.
Yeah, right.
Yes. So in our analysis, we showed that AD is highly significantly negatively correlated with education attainment and cognitive function. But honestly, those signals can also be identified using LD score regression. Right. It's just at that time, those data sets haven't been available yet. It's a recent GWAS. Yes.
So I think the problem with doing this with the neurodegeneration, neurodegenerative disorders, is that many of them don't have big enough GWAS, right? I mean,
that's right.
Your analysis is more powerful now than, but in the LD score regression analysis in the past, you haven't seen very much because
exactly.
The samples weren't big enough.
Exactly, I completely agree. I think it's not part of this paper, but they have a recent preprint that focuses on AD and PD. And they were not able to see any genetic correlation between AD and PD. But our point is that maybe it's because the genetic architecture is not that polygenic. If you focus on specific annotations, it may boost your statistical power. For example, predictive functional region or immune-related functional genome. Yes. Right? So, yes. Okay. All right. So, so that's about partition, heritability, and genetic covariance. So these are to demonstrate why functional annotation may be important for understanding genetic architecture. Now I'll talk a little bit about Gingival analysis. It's kind of a revisit Tim's point early on. So he has shown this figure. It's a predict scan. The idea of predict scan or this type of gene level analysis is that you have to impute gene expression using external data set. And by the way, this is tissue specific. You just focus on, for example, brain tissue and train this model. And then you go back to your GWAS and apply this prediction model to your data set where only genotype information was measured. Then you get imputed gene expression in the brain tissue, and then you can do case control analysis, or for quantitative traits, just linear regression, to study the association between genes and the diseases. That's the basic idea, right? So, but there are problems. So to me, to get some, I mean, they put this into this nice interpretable prediction framework, but to me, this is not too dissimilar to something called colocalization analysis. So basically, after doing GWAS, you will get summary stats for the particular trait you're interested in. But when you look at the summary stats for EQTL analysis, basically you just replace the traits into the expression of a particular gene. Then if your GWAS hit turns out to be an important EQTL for a nearby gene, then you tend to get a more significant p-value for that gene in predict scan. So that's really the intuition. But there are all sorts of problems. For example, when you see, I mean, this is too clear, right? But in practice, you will see messier signals. But even if when you see this, can you confidently say that it is the same SNP that's driving both EQTL, both gene expression and coronary artery disease in this case? Or are there two SNPs, one is driving each one of these, and then it turns out to be in LD? I think it's a non-trivial question, and it's very difficult to solve. And there are other issues. So if you have ever had any experience in analyzing data using predict scan, you tend to identify significant genes. But if you look at, you want to see which tissue is this signal coming from, you will see something like important gene, the signal come from brain, but also from skin, from gut, from all sorts of seemingly irrelevant tissues. And that's because, I mean, in a very recent GTEx paper, they showed that with limited sample size, almost all the EQTL you identify are cross-tissue EQTLs. When you increase the sample size, you get more and more tissue-specific EQTLs. But GTEx only has 800 or 900 samples at this moment. So this also tells you that cross-tissue EQTLs have stronger effect size. So when you do predict scan, it's not immune to this kind of artifact because the most predictive SNPs will be EQTL across many different tissues. So if you want to use transcriptomic information to help you figure out what's the relevant tissue for this specific locus, it's not as effective as what I just showed you before, which uses epigenetic data. Does it make sense? Okay.
So how likely is it that the chromatin structure is changing with aging and disease and thereby regulating the access of that machinery to the SNPs to regulate gene expression in the first place?
I think that's a very specific hypothesis. I can't answer that. I don't think anyone has. Oh, Alison has an answer.
I mean, I think it's certainly possible, right? That part of that contribution of aging could be epigenetic differences.
Well, I do think it's interesting that these models, even the EQTL models, don't account for this, right? Like, we don't have, does an EQTL association change over the course of aging? We don't know the answer to that.
No, we don't, that's right.
That's really important.
We know aging. I don't know whether there's any age data on any of this EQTL data.
Yeah, I think GSAC has it, but I don't think they've looked at it that way, right?
I don't think it's part of the current release, though.
Yeah, it's covariate. Okay. And same with sex. Like,
Yeah,
we're
right.
not making sex-specific EQTLs.
Or
We're
race.
not imputing them. Who knows?
Mm-hmm.
It's
Yep.
a big problem.
So there are certainly many open questions in this type of analysis, so just keep in mind. Even if you will see some wording like causal relationship in predict scan papers, it's not necessarily the case. So there are still issues.
How come it can't be more than one SNP, especially if it's a region, like a regulatory region where there's like a complex, like there's a, you could just imagine SNPs in this area affecting like the affinity or the binding, all sorts of things. And so like,
So,
one of us could have this and one of us could have that and
so
whatever.
transcription factor complex can have multiple binding sites, for example, in that region. So that's one naive example. But there are other cases as well. For example, maybe the truly functional spot is not even included in your data set. You're just tagging it using LD. Multiple SNPs are significant, but none of them may be functional. So it's actually more complex than what you have said.
So each one of those red dots above that line
Could be functional, or maybe none of them are functional. They are just in LD with a truly functional SNP.
or 20 and 20.
Or it could be a haplotype, right?
Yeah.
Of the SNP,
Yes.
sorry, to give-
Absolutely.
But from this, you can't do that.
You can't do that by just looking at this. It has to be more careful analysis.
So then you have to go in and look at it individual by individual basis.
Yeah, you can do that. I mean, if it's GWAS data, then all you have is imputed genotype, right? But if it's sequencing, it's possible. Okay. No problem. Yeah, I'm about to end this. So, okay. Yeah, I talked to Tim about this. I think he also mentioned a little bit in that talk. So, in my opinion, because of this phenomenon, it's very difficult to infer which tissue is relevant with the disease using transcriptomic data. So maybe in predict scan analysis, the point should not be to infer tissue specificity, and maybe you want to combine signals across all tissues to get a unified metric that quantifies the overall signal. And another side advantage of this is that maybe the truly causal tissue, like microglia, may not be included in GTEx, right? But you'll have some cell types that's kind of in correlation with microglia. By combining all of them together, you may essentially capture some of signals in microglia. So the way I think of this problem, I mean, I've never seen people solving this problem in the literature, but to me, it's not too different from burden test at the SNP level, right? By doing burden tests, essentially what you're trying to do is you get a z-score for each SNP, and Tim talked about how do we combine them into a single metric that represents the underlying gene, right? And he talked about Vegas. It's a simple approach. You ignore the LD, simply add up the chi-square statistics, but use permutation to get a null distribution of a test statistic. That's computationally intensive. But if you know the LD structure, which can be externally estimated, you can do better. There are more advanced statistical methods. The most recent one is called generalized hierarchy criticism test. Another one is called generalized Burke-Jones test that help you do this. Basically, you have the marginal test statistic and also LD. How do you summarize the information into a single metric? So on one level, at the single tissue level, predict scan is kind of like that, right? The difference is just now you're kind of weighting different SNPs using whether or not it's an EQTL. But at the cross-tissue level, it can also be exactly like that. In this case, it's just you have to know the correlation between different tissues, and you have the single tissue summary statistics using exactly the same idea. You can combine them together, right? Why do we know the correlation between different tissues? It's because the predicted gene expression is completely determined by your training model, right? You already trained that. You have all the weight all the weights for each SNPs in each tissue. And you also know the SNP level correlation, which is LD. So you know the correlation between SNPs. You know how SNPs determine predicted expression. Essentially, you know the correlation between between predicted expression across different tissues. So you can do this better. Maybe it's a work group topic. We can talk about that later. Yeah. So the final application I'm going to talk about is effect size estimation and risk prediction. So remember this kind of figure, right? So basically, we are able to connect complex traits with tissue and cell types by doing this sort of enrichment analysis. So these are 3 examples for immune diseases: rheumatoid arthritis, celiac disease, and Crohn's disease. And Crohn's disease and celiac disease all have a well-established gastrointestinal component, but RA does not. So we actually see that in our data. So all 3 diseases, first of all, they are significantly enriched in general functional region. They are significantly enriched in blood functional region, but only celiac and Crohn's disease are enriched in GI functional region, but RA was not. Okay? But what does this tell you? What this tells you is that now you have a SNP, okay, that's in blood-specific functional region. You kind of have this prior information that this is going to be more likely to be functional for all 3 diseases. But when you have a SNP that's in GI functional region, you know it's more likely to be functional specifically for these 2 diseases, but not for RA. So essentially, by using this enrichment analysis, you can put together this informative prior that in a Bayesian framework, you can re-estimate SNP's effect size. I think that's the point Catherine brought up yesterday. So if a gene is located in multiple pathways, of course, the prior should be higher. Here, the difference is just our annotation is at the SNP level. It's not at the pathway level because it's non-trivial to assign SNP to genes. Okay? So, but the idea is really similar. So we did that. We put together this framework. So using our informative prior, if you classify SNPs into high prior and low prior and move on to a completely independent cohort to look at the SNP's effect size, you're able to see that SNPs with high prior turns out to be more significant in an independent cohort, and the effect directions are also more consistent between the first and second cohort. So not just the absolute value of effect size is bigger, but also the effect direction is more consistent. So that's a demonstration that this kind of informative prior is indeed informative. So using that in a Bayesian framework, you re-estimate all the effect size and put that into a polygenic risk score model, which is quite straightforward. It's just a linear combination of genotype and the re-estimated effect size. Turns out we compared with multiple polygenic risk prediction models, including just a standard polygenic risk score based on significant SNPs, based on all SNPs, based on certain variable threshold, and LD-PRED. We are able to improve the risk prediction accuracy across 5 different diseases. And the reason why I only apply to 5 diseases is that it's actually not that easy to get individual level testing data that's completely independent from the training data you use in the GWAS summary statistics. So these are the ones that we found, and turns out the pattern is pretty consistent. Okay, so and we have further extended the model to incorporate multiple GWAS because we talked about genetic correlation. Turns out if you focus on type 1 diabetes, and the x-axis is the genetic correlation between type 2 diabetes versus different diseases, and the y-axis is the prediction accuracy for type 2 diabetes, you see a linear trend. So basically, if you incorporate a genetic disease that's very genetically correlated with type 2 diabetes, and you jointly model those 2 diseases, you get a higher improvement in prediction accuracy. So, so this one has been published. Yep, so that's the basic idea. So summary. So we have talked a lot of different types of annotation. So when someone mentioned functional annotation to you, maybe you were just thinking about functional prediction tools. But to me, it's really equal to external information. You have different type of raw data, including conservation, epigenetic data, and transcriptomic data. And of course, there are computational tools that help you predict whether or not a SNP is functional. But my experience is that all those tools, their results don't overlap. So it's very difficult to tell which one is doing the best job. And also, you have quantitative trait loci. If you combine genotype data versus some other molecular traits, such as expression, splicing, or proteomic level quantification, you can identify which SNPs are associated with those traits. This is another type of annotation for SNPs. And you can use allele frequency. I mean, because functional SNPs tend to be rare. Depending on the trait you study, this may be useful. You can also use LD. LD is nemesis in GWAS downstream analysis, but it also gives you information, because when you only have summary statistics, which are marginally estimated, if you have external panel that can give you LD information, I think I've demonstrated that that could be useful in several different applications. And additional GWAS for different traits may be useful, because those traits may share certain genetic components, and with different GWAS results, essentially you indirectly boost your sample size in your GWAS, right? And the one that I didn't talk too much about is chromatin interaction. So basically people have this technology to identify physical interaction between physical locations in the genome, and also computational tools have been identified to predict those kind of things. And I think in the near future, these tools may turn out to be useful. And for application of these annotations, I talked about file mapping, partition heritability, and identify relevant tissue for GWAS, and partition genetic covariance, and also gene level association test, and also a little bit about effect size estimation and risk prediction. So that's what I have for today. Thank you so much for your attention.